This video is about eukaryotic transcription. What is transcription? It is the process by which the information in a strand of DNA is copied into a new molecule of RNA. So the information in the DNA is copied into a new molecule of RNA. This process is known as transcription, and it is the first step of gene expression, and in which a particular segment of DNA is copied into RNA, and the enzyme used is RNA polymerase. So the transcription is the process by which the information in a DNA strand or a particular segment of DNA is copied into RNA molecule, a new RNA molecule, by the enzyme RNA polymerase, and it results in a complementary antiparallel RNA, and that is called primary transcript. So from the transcription we get a complementary antiparallel RNA that is known as primary transcript. Transcription occurs in eukaryotes in a way that is similar to prokaryotes. That is, transcription in eukaryotes also similar that of prokaryotes, but some differences are there. The major differences are: initiation is more complex in the eukaryotic transcription, and termination does not involve stem loop structure. In the case of prokaryotes, there is a stem loop structure in the termination stage, but in the case of eukaryotes, the termination does not need the stem loop structures. Then third one is, the transcription is carried out by three enzymes. They are RNA polymerase 1, 2 and 3. In the case of prokaryotes, there is only one RNA polymerase, but in eukaryotes there are three RNA polymerases, 1, 2 and 3. And the regulation of transcription is more extensive than prokaryotes. These are the main differences between the prokaryotic and eukaryotic transcription. That is, initiation is more complex, termination does not involve stem loop structure, and the transcription is carried out by three enzyme RNA polymerase 1, 2 and 3, and the regulation is more extensive than prokaryotes. Now coming to the enzymes involved in eukaryotic transcription. Unlike prokaryotes, where all RNA are synthesized by a single RNA polymerase that we discussed earlier, but in the case of eukaryotes, there are three RNA polymerases. They are responsible for transcribing different types of RNA. So there are three different RNA polymerases are used in the eukaryotes, and they transcribe different types of RNA. First one is RNA polymerase 1, which is located in the nucleolus. And transcribe 28S, 18S and 5.8S rRNAs. 28S, 18S and 5.8S rRNA genes. Next one is RNA polymerase 2, which is located in the nucleoplasm and transcribe the protein coding genes to give pre-mRNA, and also genes encoding small nucleolar RNAs, snRNA. They are involved in the RNA processing, rRNA processing, and small nuclear RNAs, snRNAs, they are involved in the mRNA processing. So the RNA polymerase 2 located in the nucleoplasm and transcribe the protein coding genes to give pre-mRNA, and also gene coding small nuclear RNAs, nucleolar RNAs, and they are involved in the RNA processing, and small nuclear RNAs, they are involved in the mRNA processing. Next one is RNA polymerase 3, which is also located in the nucleoplasm and transcribe the genes for tRNA, 5S rRNA, U6 snRNA and 7S RNA. So the RNA polymerase 3, which is also located in the nucleoplasm and transcribe the genes for tRNA, 5S rRNA and U6 snRNA and 7S RNA. Each of these three eukaryotic RNA polymerases contain 12 or more subunits. So they are large complex enzyme. All these three contain 12 or more subunits and they are large complex enzymes. Now let us discuss the features of eukaryotic transcription. Transcription in eukaryotes occurs within the nucleus and the mRNA produced moves out of the nucleus into the cytoplasm for translation. That is, the transcription takes place in the nucleus and the formed mRNA or primary transcript move out of the nucleus into the cytoplasm for translation. Then the initiation of RNA synthesis by RNA polymerase is directed by the presence of a promoter site on the 5' side of the transcriptional start site. That is, if this is the DNA strand to be transcribed, this is the 5' end, this is the 3' end. If this is the transcription start site, there will be a promoter site on this 5' end of this DNA strand. And the initiation of RNA synthesis by the RNA polymerase is directed by the presence of this promoter site on the 5' end of the transcriptional start site. Next feature is the RNA polymerase transcribe one strand, the antisense strand of the DNA template. That is, if this is the DNA strand, here only one strand acts as the template, and that template strand is known as antisense strand or the non-coding strand. And the RNA synthesized is the complementary of this strand. So the strand which are used as the template is known as antisense strand or non-coding strand. Then RNA synthesis does not require primer. In the case of replication, the DNA polymerase require a primer to start the replication. But in the case of transcription, RNA polymerase does not need a primer. And the RNA synthesis occurs in the 5' to 3' direction with RNA polymerase, catalyzing a nucleophilic attack by the 3' OH of the growing RNA chain on the alpha phosphorus atom on an incoming ribonucleoside 5' triphosphate. That is, synthesis is always in the 5' to 3' direction, and it is catalyzed by the enzyme RNA polymerase, and there in the growing end there will be a nucleophilic attack 3' OH of the growing RNA chain on 5' phosphate of the incoming ribonucleoside 5' triphosphate. So there is a nucleophilic attack between the 3' OH of the growing end and the alpha phosphate of the incoming nucleoside triphosphate. And the newly synthesized RNA, known as the primary transcript, which undergoes processing to become maturation. So the mRNA in eukaryotes is processed from the primary RNA transcript. The process is called maturation. Next is the process of eukaryotic transcription. Eukaryotic transcription. What are the processes inside the transcription? The basic mechanism of RNA synthesis by this eukaryotic RNA polymerase can be divided into the following phases. They are initiation, elongation, and termination. There are 3 different steps in the eukaryotic transcription. They are initiation, elongation, and termination. First one is initiation. During initiation, RNA polymerase recognizes a specific site on the DNA upstream from the gene that will be transcribed, called a promoter site, and then unwinds the DNA locally. Consider this is the DNA double helix. In this DNA double helix, one strand to be transcribed. So, before starting the transcription, the RNA polymerase which recognizes a specific site, that is a promoter site, which is present in the upstream end of the DNA to be transcribed. So first the RNA polymerase recognizes that specific site known as promoter site, then binds to the promoter site and then unwinds the DNA locally. And the promoter site for RNA polymerase II include a highly conserved sequence. They located about 25 to 35 base pair upstream to the 5' end of the start site, which has consensus TA-TA-AT-A-AT region and is called Tata box. That is, the promoter site contain a 25 to 35 base pair sequence, consensus sequence known as Tata box. Here we discussed that here is the promoter site. So in this promoter there will be a 25 to 35 base pair sequence that is known as Tata box, and that sequences are TA-TA-AT-A-AT and this is called Tata box. Since the start site is denoted as position +1, the Tata box position is said to be located about position -25. That is, consider that this is the start site and we can denote it as +1 site. So here this will be -1, -2, -3 like this. So the sequences of the promoter site, that the promoter site contains the Tata box, that will be referred to as -25 region, or that will be -25 sequences or base pairs upstream to the 5' end. That is, this is the +1 end, then -1, -2 like this. Here at -25 there will be the Tata box, and that sequences are known as TA-TA-AT-A-AT and is called Tata box. And this Tata box resembles -10 sequence in prokaryotes. At sequence is TA-TA-AT, except that is located further upstream. So that is, in prokaryotes there will be a -10 sequence, but in eukaryotes it will be -25 sequence. Both elements have essentially the same function, that is recognition by the RNA polymerase in order to position the enzyme at the correct location to initiate the transcription. So both the Tata box in eukaryotes and prokaryotes have similar function, that is the recognition by the RNA polymerase in order to position the enzyme at the correct location to initiate the transcription. The sequence of around the Tata box, also important, that is it influences the efficiency of initiation, and the transcription is also regulated by the upstream control elements that lie 5' to the Tata box. That is, the transcription is regulated by upstream control elements that lie 5' to the Tata box. And in order to initiate the transcription, RNA polymerase II requires the assistance of several proteins. RNA polymerase II requires the assistance of several proteins or protein complexes. They are called transcription factors, which they must be assembled complex on the promoter in order to start the transcription. And they have the general name transcription factors 2. 2 means it is for the RNA polymerase II. So the transcription factor 2, it is the generic name of that proteins. Now coming to the initiation process. The first event in initiation is the binding of the transcription factor 2D protein complex to the Tata box via one of its subunit called TBP, that is Tata box binding protein. This is the initiation phase. Here you can see Tata box -30. This is the promoter region. You can see the promoter region. And this is the +1 start site. So the RNA polymerase bind to this side. So before that, the first event is the binding of the TF2D along with the TBP, Tata box binding protein. So first the TF2D binds at the Tata box side along with the Tata binding protein. As soon as the TF2D complex has bound, then TF2A binds and stabilizes the TF2D Tata box interaction. And then next TF2B binds to TF2D. That is, as soon as the TF2D TBP binding, then TF2A binds. TF2A binds to stabilize the interaction between Tata box and TF2D. Then TF2B come and binds. TF2B can also bind to RNA polymerase 2. So act as a bridging protein. That is, the TF2B also bind to the RNA polymerase 2 and act as a bridging protein. Then the RNA polymerase 2, which has already complexed with the TF2F, now binds. That is, in this stage, when first stage TF2D and TBP binds, then TF2A come and bind, then TF2B come and binds, then RNA polymerase 2 along with the TF2F binds with this, and the RNA polymerase 2 also binds with the TF2B. So this is a bridging protein, TF2B. Then TF2E and TF2H come and bind. So in the first step, TF2D along with the TBP, Tata box binding protein, come and bind to the Tata box region. Then TF2A come to stabilize the interaction between TF2D and Tata box. Then TF2B come and binds, and TF2B is always bind with the RNA polymerase 2 and which act as a bridging protein. Then RNA polymerase 2 along with the TF2F come and bind, then followed by TF2E and TF2H come and bind. And this final protein complex which contain at least 40 polypeptides, and they are called transcription initiation complex. Transcription initiation complex. That is the complex known as transcription initiation complex, and it contains 40 polypeptides. Next is the elongation phase. We earlier discussed that TF2H, which already bind with the RNA polymerase 2, and this TF2H has two functions. First one is it is a helicase function. It has a helicase function and it requires ATP to unwind the DNA helix and allowing transcription to begin. So TF2H has the helicase activity. Helicase means which unwinds the DNA. If this DNA is like this, this helicase will unwind the DNA like this. The next function is it phosphorylate RNA polymerase 2. The RNA polymerase 2 is phosphorylated at the C-terminal tail called the C-terminal domain, CTD of the RNA polymerase 2. So this protein TF2H will polymerize or phosphorylate the C-terminal domain of the RNA polymerase 2, so that this enzyme can change its conformation and dissociate from other proteins in the initiation complex. We discussed that the RNA polymerase II start the transcription when the C-terminal domain of the RNA polymerase II is phosphorylated by TF2H. So it has two functions. One is helicase function and the other one is phosphorylation of the RNA polymerase II. And only the RNA polymerase II that has non-phosphorylated CTD can initiate transcription. But only an RNA polymerase II with phosphorylated CTD can elongate RNA. So the transcription can start with the RNA polymerase II with non-phosphorylated CTD. But for the elongation of the RNA polymerase II required phosphorylated form. Phosphorylated CTD, C-terminal domain. When it gets phosphorylated, the RNA polymerase II starts moving along the DNA template and synthesizing the RNA. That is, the process enters the elongation phase. So, when the CTD is phosphorylated, the RNA polymerase starts to move, and that phase is known as elongation phase. And the RNA synthesis occurs in the 5'3' direction with the RNA polymerase catalyzing a nucleophilic attack by 3'OH of the growing RNA chain on the alpha phosphorus atom on the incoming ribonucleoside 5-phosphate. If this is the growing RNA chain, here is the 3'OH. Here will be a nucleophilic attack between the 5' nucleoside triphosphate and the 3'OH of the growing RNA chain. The RNA molecule made from protein coding genes by RNA polymerase II is called a primary transcript. So the RNA synthesized from this transcription process is known as primary transcript. Next is termination phase. The nucleotides are added by the RNA polymerase until a termination occurs. That is, the elongation of the RNA chain continues until the termination occurs. Unlike the RNA polymerase in prokaryotes, RNA polymerase II does not terminate the transcription at a specific site, but rather transcription can stop at varying distances downstream of the gene. That is, in the case of prokaryotes, there will be transcription termination signals, or there will be some raw factor, or there will be some hairpins like structures to terminate. In this case, there will be no transcription termination signals. So the transcription can stop at a varying distance downstream of the gene. That is, if this is the gene to be required, the transcription starts varying distance downstream of this gene. So the transcription is continuous. That is, the RNA genes transcribed by RNA polymerase II lack any specific signals or sequences that direct the RNA polymerase II to terminate at a specific location. So it will continue. The RNA polymerase II can continue to transcribe RNA anywhere from a few base pair to thousands of base pair past the actual end of the gene. If this is the gene, this is the actual end of the gene, the RNA polymerase again transcribe the gene that does not required. So the transcript is cleaved at an internal site before the RNA polymerase II finish transcribing. So that transcript, if this is the DNA, the RNA polymerase, this is the RNA polymerase which moves. So the primary transcript is synthesized like this. If this is the end of the DNA to be transcribed, again the RNA polymerase will transcribe the RNA, and at internal positions it will be cleaved, and this released RNA is called as primary transcript. And that cleavage site is considered as the end of the gene. And the remainder of the transcript is digested by 5' exonuclease. That is, if this is the gene to be transcribed, this is the original end of the gene to be transcribed, then all these extra transcript will digested by 5' exonuclease. This is cleaved by the 5' exonuclease. And when the 5' exonuclease catches up to RNA polymerase II by digesting away all the overhanging RNAs, it helps disengage the polymerase from its DNA template strand and finally terminating the round of transcription. That is, when the exonuclease starts to function, it digests all the overhanging RNA and help the polymerase to disengage the polymerase from its DNA template and to release from the DNA template, so that the transcription can stop. So we get a primary transcript and it is not active. To get a mature mRNA, we want to process it. So let us discuss the processing of the mRNA. That is known as RNA processing. The primary eukaryotic mRNA transcript is much longer and localized into the nucleus when it is called heterogeneous nuclear RNA or hnRNA or pre-mRNA. The first formed mRNA transcript is known as pre-mRNA. It undergoes various processing steps to change into mature RNA that we will discuss. In the processing, the first one is cleavage. The larger RNA precursors are cleaved to form smaller RNAs. That is, we get a large RNA transcript. It will cleave into smaller RNA fragments. The primary transcript is cleaved by ribonuclease phosphate. Primary transcript cleaved by ribonuclease phosphate to form 5 to 7 tRNA precursors. That is, to form 5 to 7 tRNA precursors. Next process is capping and tailing. Initially at the 5' end, a cap which is known as 7-methyl-guanine and a tail of poly A at the 3' end is added. That is, for this primary transcript, the 5' end is capped by 7-methyl-guanosine and at the 3' end poly A tail is added. This is capping and tailing, and the cap is chemically modified molecule of guanosine triphosphate. Next one is splicing. Eukaryotic primary transcript mRNA, made up of two types of segments. They are non-coding introns and coding exons. Non-coding introns and coding exons. That is, this is exon, this one intron, then this is exon, then this one intron. So the primary transcript contain non-coding introns. This is the non-coding introns and coding exons. These are the coding exons. So the introns are removed by a process called RNA splicing, where ATP used to cut the RNA, releasing the introns and joining two adjacent exons to produce mature mRNA. That is, these introns are cleaved by RNA splicing. For that, ATP is utilized and these exons are joined to produce the mature mRNA. So we get exon 1, exon 2. This is first exon, this is second exon, and the introns are removed. This type of RNA is called as mature mRNA. Next one is nucleotide modification. They are most common in tRNA methylation, that is methyl cytosine, methyl guanosine, or deamination of inosine from adenine, dihydrouracil, pseudouracil, etc. So the post-transcription processing is required to convert primary transcript to functional RNA. Now coming to the significance. The transcription of DNA is the method for regulating gene expression, that it occurs in preparation for and is necessary for protein translation. So it is necessary for the translation of protein because from the mRNA the protein is synthesized. These are all about transcription in eukaryotes. Thank you.